Okay, thank you very much. Can you hear me? Good. I think it was the longest introduction I've ever got to a talk. But unfortunately, I could follow part of it. And we will see if I have time to talk a little bit about the Kampfläufer, which is a fascinating story. But the main thing today will be talking about the domestic animals and how they became domestic animals. And I call this a walk in the footsteps of Charles Darwin. And I will explain why this is a walk in the footsteps of Charles Darwin. But let's start by this image here, which I think is really lovely. So this is in fact an illustration from a children's book in Sweden about the origin of our domestic animals. So to tell kids where is our domestic animals coming from. And this is the red jungle fowl, which are the wild ancestor, which occurs in Southeast Asia in the jungles there. And these are the different types of domestic chickens. And you could see here this wonderful biological diversity among our domestic animals. And you could say, this is what I've been exploiting in my research for the last 30 years or so. So about Charles Darwin. So Charles Darwin is, of course, the most famous biologist together with Linnaeus, perhaps, which is from Uppsala. And so he's famous for his theory on the origin of species. And the thing is that during the voyage of Beagle, he came to the Galapagos. So this is the Galapagos, the Darwin's finches on Galapagos. And in fact, these are the different species that we have been sequencing now and done several types of studies on them, and we were there very recently. So Charles Darwin came to the Galapagos he arrived on September 17, 1835. So in fact, September 17 is my birthday, so it's a link there. But he came back to England 1836. So then it took him 23 years before he published the origin of species. And you could see already from what he writes about his voyage that he has this idea about evolution already. But he waited 20 years to publish. You know, this would not work today. As a scientist, you need to publish to get grants and so on. But he had money from his family, so he could study. And so the thing with me is that I was trained as a biologist. So as a kid, I was very interested in birds and animals and wildlife. And I wanted to study biology. I did my first work on Atlantic herring, in fact, in the late 70s. But then I decided to look for another position. I got a position at Agriculture University. I never planned to work with domestic animals. But I was living in Stockholm and I commuted to Uppsala, which it takes like 45, 50 minutes. So I was doing that every day for 2 years. And this is in the early 80s, so there's no computers so you could carry on, so to speak. So you could read. So I was reading, and one of the books I was reading during my trip was this book, The Origin of Species. And what was fascinating to me was that the first chapter of the origin of species is about domestic animals and domesticated plants. So what Darwin did, he used the domestic animals and domestic plants as a proof of principle for the theory of natural selection, how natural selection causes evolution. Because what he's saying is, so what he say in this book is that this book is one long argument. So what Charles Darwin was doing during this 20 years or more before he published, he collected evidence because he understood how controversial the theory of the origin of species is in a time because all our religions has these ideas about how humans occurred on Earth and how biodiversity evolved, or not evolved, but was created, you say. And he formed this scientific theory, how it could evolve by natural selection. And he wanted to have a very, very strong argument. And he used domestic animals as a proof of principle. So he published, 9 years after publishing The Origin of Species, he published these 2 volumes of the variation of animals and plants and the domestication. So where he, for instance, compared the wild boar with the domestic pig, what type of changes have happened. And he was doing breeding experiments himself with pigeons. And he could show that he could change their phenotype, their appearance, by selective breeding. So there was the proof that selection changed the phenotype and can cause evolution, so to speak. And what he writes in the Origin of Species is there's no reason why these principles that have worked so efficiently in the domesticated species should not be applicable also in nature. That is what he says. And I was inspired by that, and as a biologist, and I also felt at that time, this is early 80s, that domestic animals were investigated as a biological in biology. So the thing with our domestic animals is that they have evolved by what we call selective breeding during thousands of years. So for the last 10,000 years, humans have interacted with our domestic animals and selected the ones that could serve purposes for us, either as transportation or production of milk or meat and so on. So to illustrate, so this is a picture I took when I was like 14 years old. So I got a camera. So this is in fact my uncle taking his cattle home for milking in the afternoon on a farm in south of Stockholm. So before we get into it, I would like to mention that I would say there is two types of selection which have occurred during the last 10,000 years. So one is cherry picking. And what I mean by cherry picking is that it's mutation with large visible effects. So this is things we have observed over domestic animals. And I take this as an example as a cherry picking. So these are these white horses, which are being selected because we think they are so beautiful. So the first time these are mentioned, to the best of my knowledge, is Herodotus, the Greek historian, which tells that the Persian emperor Xerxes brought holy white horses when they were attacking Greece. This is about 2,500 years ago. If you go to medieval time in Europe, all European countries are full of white horses, and they're often used by the royalties because they are a symbol for beauty and power and so on. So this is in fact the Swedish king, Carlos XI, and you use that on the poster for this symposium. So this is at the Battle of Lund. And if you come to Sweden, you could go to the castle of Gripsholm, which is southwest of Stockholm, and you will find this portrait here. The name of the horse is Brilliant, in fact. Sweden won the battle, in fact, and that was set the border between Denmark and Sweden. So. But then we have these gradual changes also. So over time, we have selected our domestic animals gradually to increase their size or decrease size, increase the size of eggs and so on. And this is another type of mutations. They could have tiny, small, or medium effects, so much milder effects. So illustrated by these are selection lines in chickens. So these chickens came from the same origin. This is 1957, in fact. And in every generation, this person that did this breeding experiment has selected those for high growth in one line and for low growth in another line. So after 40 generations of selection, there is about almost 10-fold difference in body weight. And this has been done without any knowledge about which genes there are. It's just by selecting, measuring the body weight, and allowing in this line the most heavy birds to breed, and this line the one which has lowest weight. And here this selection response is the effect of hundreds of genes which have contributed, each with a quite small effect. So very different compared with this cherry picking, which is when we see something, we like it, and we keep those animals. So this is an illustration of what I've been working on over the last 30 years. So we have studied all types of domestic animals. So we have crossed the wild boar with the domestic pig, in this case, and studied the segregation of genes in relation to phenotypes. These are the selection lines. We have also studied dog genetics, horse genetics, and chicken. We crossed the wild chicken with the domestic chicken. So a lot of the variation you see on this picture, we have in fact nailed down to the gene and the mutation underlying this. So I could have spent my whole talk on a few of these examples here. So for instance, how the pigs become white, for instance. Many of the domestic pigs we use today is white, and they're white because they have a mutation in a gene that is critical for the pigment cells when they migrate during embryonic development out into the skin. So they lack pigment cells in the skin and hair follicle, and that is the reason why they are white. But I can't talk about all the things we have done, so I have selected 3 discoveries which I think is illustrative and interesting to see what have happened with our domestic animals. So the first example I will give is this discovery why domestic pigs develop more meat and store less fat than wild boars. So this is from our cross between the wild boar and the domestic pig. So we started this experiment in the late 80s. So I moved to Uppsala in 1980 and started to do work on domestic animals in the beginning of the 80s. And what I realized in the early 80s was that this was the time when the technology of molecular genetics was becoming available. So what I realized was that the new possibility to study the DNA would make it much more interesting to study the genetics of domestic animals, because we can go into the gene and see what genetic change ever happened. So that is what I mean by walking in the footsteps of Charles Darwin. So Charles Darwin used the domestic animals as proof of principle that selection will change the phenotype. My view was that with the DNA technology, we should be able to see what genetic changes have happened, and that's what we have been doing. So what you see here is, in fact, so we crossed this one. So this is the first hybrid generation, and this is the second generation. So these F1s, they look all the same. So all of these get 1 chromosome from the wild boar and 1 from the domestic pig. So they're all very similar. What happens from F1 to F2 is that you get Mendelian segregation. So these are 50% wild boar, 50% domestic on average. But if you look at an individual gene, they could either be homozygous, as we say, they get both chromosomes from the wild boar or both from the domestic, or heterozygous. And that's exactly what you see here, illustrated by this variation in pigmentation. So you see this one has got the genes for the coat color of the wild boars. These have what these guys have, and these have combination of genes that you don't find in the parental line, so to speak. So this just illustrates this variation in genes going on in this. But these segregate for all type of genes that have been selected during domestication. And now I come back to this illustration by Charles Darwin. So what is interesting here is that this is in fact the same breed as this breed. It's called the large white. You could see Yorkshire large breed here. That is the same. This is in England in the mid-1800s. This is in Sweden in the late 20th century. And what is the difference between these guys? Well, this is a fat pig. You could say basically before Second World War, and in the 1800s and earlier, the breeding goal was to have fat pigs, because the problem was not to get obese and have cardiovascular problems and so on. The problem was to survive, to get sufficient energy to work hard in agriculture or in industry. So people wanted to have the fat. But that changed completely, basically after Second World War, when our lifestyle changed with cars and so on. So from the 50s, there's been for every year there's been selection going on to reduce fat deposition and increase muscle mass, because it's the protein that the consumer pays for. They don't pay anything basically for the fat. So this is what have happened, that we have. So if you will take these pork chops and if you start from 1900 to today, you will see that this fat layer become thinner and thinner, and it become more and more muscle meat, so to speak. And what we showed in this study was that the most important single factor was a gene called IGF-2, which stands for insulin-like growth factor 2. So one of the known growth factor in mammals. And we could show that depending if the pig got the gene variant from domestic, they get 4 kilograms more meat and a bit less fat. So if you think a farmer that has pigs, if his farm is full of the one with the mutant, he gets 4 kilograms more meat from every pig without feeding them more. So today, basically all production in the Western world, America, Europe, Australia, even China today, is based on pigs carrying this specific gene variant that I will tell you about. So I used to say that this is part of our, you could say, food security today, that we produce food with less resources. So and we were able to nail this down to the specific mutation. So this is the IGF-2 gene. So genes have exons and introns. Exons code for the protein sequence. So 1, 2, 3, 4, 5 is coding exons, and whereas introns doesn't code for anything. They cut out when the protein should be produced. So what we were able to show was that there was the critical mutation is located in this intron 3 here. So this up here is the sequence of a region here in this intron in the wild boar. So this is the sequence CG, CD, T, CD. And then you come to this G here. And dot here means that this is the domestic pig. And you could see that this is, a dot means that it's identical. So the domestic is identical except at this position here, where G has changed to A. So then you should remember that the genome of a mammal is 3 billion base pairs, about the same in pigs as in humans. So out of those 3 billion, if you change that G to an A, the pig gets much more muscle. And it occurs in an intron. But so why is that? So the first indication that this was an important finding was when we compared this sequence with other species. So here you have cattle, horse, dog, rabbit. We could go back to rabbit later in the talk, but human, mouse, rat. And look here now. So these are sequence differences. So if you take this, these are the sequence difference between the pig and the human. But look at this block here. It's completely conserved among all the placental mammals. We have at least 20 different species of mammals. They're all identical over this sequence. What that tells us is this must be an important sequence. So why should a mutation in an intron has this effect? Well, this comes to transcriptional regulation. So you have the genes. So in this cartoon, red bars here are what becomes the coding sequence of this gene. And you produce mRNA, which is just this coding part. But how much? So the transcriptional regulation is where, in which tissue, when, at what stage, and how much a gene is active. And then you have a number of transcriptional factors. So these are proteins that bind the DNA and regulate the activity of this gene. So these are super critical. All cells have the same gene content, but what the liver cell do and what the brain cell do is very different. And so our suspicion was, of course, that it's one of these regulatory elements which are affected there. So these proteins, they bind DNA with a specific DNA sequence. And since this sequence is so highly conserved among mammals, our hypothesis was that it disrupts the interaction between a protein and the DNA sequence. And that was exactly what it was. So without going into the experimental details, what we could show was that in fact, so in the wild boar, as well as in humans, you have a sequence here, and G is the sequence, and that binds a protein in the nucleus, and that regulates how much IGF-2 is made. But what happened here in the domestic pigs, so this is a Chinese pig, in fact, they are like the wild boar. What happened in the domestic pigs was that a mutation happened, which changed G to A, and when that happened, this protein cannot bind anymore. And then this become partially unregulated, and you upregulate and make more of this growth factor, and that promote muscle growth. We were able to show that that protein was a protein that people had missed when they classified or studied the human genome. So we were able to name that to CBD6, and it's a transcription factor that interacts with this site. And CBD6 has a very interesting history in itself, because CBD6 has evolved from a DNA transposon. So what is a DNA transposon? Well, this is this selfish DNA that can jump around in the genome of their hosts. So Hermes is the name of a DNA transposon belonging to the same family as the origin of CBD6 was. And this is a DNA transposon occurring, it's active in the housefly. So this DNA transposon were active in the mammalian genome hundreds of million years ago. So what we showed was that this jumping DNA entered into a primitive mammal about 200 to 250 million years ago. This DNA transposon integrated into our genome. And then it was considered, I mean, you could consider at the beginning it was just junk DNA, it didn't do anything. But at some point, so when we look at the marsupials, we don't find it, but when we find remnants of it, but it's not present there. But in the Etherians is the placental mammals. So something happened during this period of time that it started to have a function in the placental mammals. Because what we now see is that all placental mammals has this transcription factor, and it's highly conserved among all placental mammals. So it contributed an important function. So what do we do if we would like to learn what the function of a gene is? So now we decided to knock out this gene. So what happened if you remove CBD6 from a wild type mouse? Well, it looks pretty similar, as you see, but it's the same picture. But in fact, they look very similar in real life also. But if you look carefully, they have more muscle, they have a larger heart, and they have a larger liver and kidney. So what we discovered is a mechanism in the placental mammals that is regulating the growth of organs. So this started with crossing the wild boar to the domestic pig, and we learned something about human biology and all the biology of placental mammals by this approach. So what we are really interested in today is to see, so what we know is that if with a pig mutation we upregulate IGF-2 expression very much, 10, 30-fold. If we knock out CBD6, so this is the pig mutation, this is the knockout mouse, we get very similar effects. We upregulate IGF-2 expression and we get this effect. So I would argue it's very likely that this mechanism also controls muscle mass in humans. So when you are trained, we think this is one of the mechanisms that is regulating muscle growth. Because muscle is this fantastic organ that adapts to how much we are using it. So this is what we are working on now, because you could think of there is something happening with the protein when we are training, or there's something happening with the DNA that disturbs what we call epigenetic changes that affect the interaction between CBD6 and the DNA here. And that is what the ongoing work is, to try to understand the mechanism in vivo. So, as I said, this mutation, when we compare the wild boar with domestic pig, this is the most important change that has affected muscle growth in the pigs. But growth is a complex trait affected by many genetic factors. So you shouldn't think that it's just one factor. This is the most important reason for difference, but there are many others contributing. And I would like to illustrate by this image, which I think is, this is chicken instead. But this illustrates what type of changes you could occur by selection. So this is a control line of chickens, what the chicken broiler lines were in 1960s. So they were kept without selection. So this, and then you give them the same feed as these guys. So this is an advanced line from 2001. And so two different genetic materials, giving in the same environment with the same feed. And this is the difference in growth rate. So you could compare day 43 with day 85. So you get much more protein with half the time. So this have had an enormous impact on food consumption. When I was a kid, we got cooked hen in school. That is not anymore because it's so cheap to produce meat from chickens these days. So you could buy. So buy chicken meat was expensive in the old time because it took a long time to produce this, and this has had an enormous impact on the consumption of chicken meat worldwide. So it goes from 10 million ton to 80 million tons from 1960 up to 2008 or what it is. So this has a huge impact on food security around the world. So even small, of course, industrial production in Europe, but also small farms in Africa can take advantage of these improved lines. You could get more meat with less feed and in a shorter time. Second example. Now I will tell you about the gatekeeper mutation in horses. So this is a single base change which increases horses' gait repertoire. So this is the illustration where we published this story. And so this is an Icelandic horse during a competition in Iceland. So this horse is pacing, in fact. So you see it moves the legs on the same side together. So the background is as follows. So all horses can do this. So they can do walk, which is a 4-beat gait, which take one step at a time. Trot is a diagonal 2-beat. That means that they put the two diagonal hooves down at the same time. Then they fly in the air and put the other two together. So 2-beat. Canter is 3-beat, and the gallop, which is the fastest, so it should be 4-beat. This is Swedish, in fact, takt. So this is the fast, and they use this with increasing speed, exactly as we do. We walk, and then we decide we are the way to miss the bus or the underground. Then we start to run, and we change the pattern of locomotion. But what you also have, you have so-called gated horses. So gated horses. So this is these are all Icelandic horses, or one Icelandic horse is doing all this type of gaits. So tölt is a special gait in Icelandic horses, which is a 4-beat gait. What is really special with this gait, so it's almost as fast as the trot, but they always have one hoof on the ground, which means that if you ride this horse, you never fly in the air, you're always grounded, which means that you have a very smooth ride. So it's like sitting in a sofa and riding. So very popular if you're not so if you're unexperienced, I recommend to use the tölt. So it's called ambling gait, and different breeds of horses have this type of ambling gait, but only the one that we call gated. But some Icelandic horses can also do the pace, which is a 2-beat gait. So now, but lateral. So this is a diagonal movement, and this is the lateral movement. And then, yeah. So if you look at the horses of the world, you could classify them as gated or wild-type horses. So for instance, you have thoroughbred horses here, you have Arabian horses and so on. So majority is in fact not gated. But then you have a number of gated horses, so Icelandic horses an example. Many in the Americas, in fact, and in Asia, and I will come back to that later. So just to give you one example of what type of funny gaits you could see in these horses. So this is the Paso Fino horse. So it's a horse from the Latin America, and they have a very special gait. So it looks like Spanish dance, I think. So this Paso Fino, the definition of that, you should be able to ride a horse, you should have a sword in one hand and a glass of wine in the other hand without spilling the wine. So different breeds have different gaits. So you could Google on this and see some fascinating gaits in different. So in different breeds, they have selected different variants of this ambling gait. But Icelandic horses, there are two types. So you have four-gaited, so they can do walk, trot, gallop, and this tölt. And then five-gaited, then they could also do this pace. So about half of the population are four-gaited and half are five-gaited. So if you're a geneticist, then you're really is challenged by this. You want to know what is the reason for that some are five-gaited and others are four-gaited. So you could, of course, ask, is there a genetic explanation for this, or is this just a question of training? So you need to train them to pace. And so how do we answer this interesting question in the golden age of genetics? I call this the golden age of genetics because all the tools we have got in order to study genetics of different species. So what we do is something called genome-wide association screen. It's exactly the same approach as if you want to study which genes are affecting diabetes in humans or any disease in humans. You do exactly this type of thing. So what you do is you collect material. So we had five-gaited horses and four-gaited horses. So we had 45-gaited, so 80 chromosomes because they are diploids, so they carried one chromosome from father, one from mother. And we had 34-gaited. And what we then do is that we screen them with genetic markers. And genetic markers are this. So you go to one position in the genome. So now we start with chromosome 1, at nucleotide position 1234. And in the horse population, there is A or G at this position. So if you sequence your own genome, you will find such a sequence variant of a nucleotide every 1,000 base pair approximately. So you are heterozygous, as we say, at approximately 3 million position in your genome because of difference between the chromosome you received from your father and your mother. So we take advantage of this, and what we do is to see, because what we are expecting, if you find such a if you have a genetic marker which occurs in the vicinity of a mutation that is controlling this phenotype, we expect to see a difference in the frequency among the two groups here. So in this case, at this position, you see there is 41, 39, 30, 30, very, very similar, not statistically significant. We say it's unlikely that there's something in the vicinity of this genetic marker. But then you go across the genome, and then you come to chromosomes. So in this case, we had 50,000 markers across the whole genome. So we come to chromosome 23, to this nucleotide position. We have A or C in the horse population. And when we look at the five-gaited horses, we find 78A and 2C, but four-gaited were equal 30-30. This is highly significant, and this is the graph illustrating that. So this is the likelihood to get this observation. So what you see here is all noise. These are the 50,000 markers, that every dot is one marker. But you see this marker tells us that in the vicinity of this position, there must be a genetic change which is critical for being five-gaited or four-gaited. Identification of the mutation. So we found that, in fact, this genetic marker was in the vicinity of the specific mutation. And there was a single base change in a gene called DMRT3, which is coding for a transcription factor that regulates the expression of other genes. So CBET6, as I mentioned before, is also a transcription factor. This is another transcription factor. So it belongs to a family of genes that has a role in sex determination. But it turns out that DMRT3 has got another function in the vertebrates. So when we compare the wild-type horse and the mutant horse, so now we compare them with the amino acid sequence. And you could see here, this is amino acid 301. There is a stop codon in the mutant. So they get a truncated protein. So the wild-type horse has this full-length protein. So this protein is about the same size in humans and in horses. But these mutant horses, they have a shorter version of that protein. And I will come back to you and tell you what this protein is doing. But let us first see the consequences. So what we can see here is that this mutation has been spread along the world. So this is the frequency of this mutation in different breeds So after discovering this mutation, we wanted to see how widespread. We discovered on Icelandic horses, which are up here. And then we contacted people around the world. So I contacted a person on Japan, and I asked him, well, do you have any horses in Japan that has these ambling gait types? And he told me, well, we have this local breed called the Hokkaido horse, and they were very popular during medieval time because the samurais were riding heavily armed, and they liked the smooth gait of these horses. And we found the same exactly the same mutation in these horses. And we could follow it in Chinese breeds, Mongolian horses, Kyrgyz horses. Going east here, you get into Europe, there are several breeds in Greece which has this mutation. We know that the Vikings brought them to Iceland. The conquistadors brought them to the Americas. So a number of the breeds in the Americas has this specific mutation, and they can make. So basically, all gaited horses in the world carry the same mutation, which probably arose somewhere in Asia some 1,000 years ago and spread. And this is what I call cherry picking. So humans could never breed for having horses doing these ambling gaits or pacing because we could never foresee that horses could do this. But humans were riding horses, and they noticed that these horses have a special gait and liked it and spread over the world. And it increased the way how we have utilized horses during the last 2,000 years. So we also found them at a high frequency in trotting horses and pacing horses used for harness racing. So these are trotters, and they are mutant. So AA is mutant here in DMT3, and these are pacers. So these are doing this. So these are both this 2-beat gait, but the diagonal and the lateral. So we were quite surprised to see that these two different types of horses have the same mutation. And what happens is that apparently you're selecting on other, what we call genetic modifiers, that allow them to either be primarily trotters or pacers. But what we think this mutation is also doing is that it's inhibiting the transition from trot to the gallop. So these horses are not allowed to do the gallop. So they run at very, very high speed without galloping. So this is our model that all horses do this. But if this mutation is permissive for doing the tölt and the pace, and it inhibits a transition from the trot or pace to the gallop. So these horses, in fact, with this mutation, they're really lovely for this ambling gait, but they are not as good for the gallop. So if you go to race horses, thoroughbreds, where you use the gallop, then we don't find the mutation. So also draft horses carrying heavy load are wild type. So depending how we use the horse, the mutation occur in different frequency. So how old is this mutation? So when we published this, we got the cover of Nature, which is always very nice for the researcher to get. And I looked for what could be the cover illustration. So I searched for pacing horses, and I came up with this thing. So this is a very famous sculpture from China. So it's a bronze. It's called the bronze pacing horse, poised on a swallow. So this is a swallow with wings outstretched. Eastern Han Dynasty, 2nd century. So then is the question: did they have pacing horses 2,000 years ago in China? I think it's very likely, but of course we can't know if the artist was really cared about how they. But there are other examples of this, and this is also very interesting. Plinius the Elder, so Plinius the Elder, he died during the eruption of Vesuvius, 79 after AD, and he described some Spanish horse as these horses have a unique leg action that differs from that of other horses. So we think these were in Europe all 2,000 years ago. So what is the function of the MLT3 and where is it expressed? So this is what I think is when genetics is really beautiful, because we use the genetics to prove that this mutation is changing the gait of horses. The next question is, of course, what does this gene do? But we discovered the gene completely without any knowledge of its biological function, pure by the fact by comparing the genetics of horses having different gaits. And the first question we ask is, of course, where in which cells is this gene expressed? If we have found out that they were expressed only in the skin, we would be very worried that we have the wrong gene. But what we found out was that it's expressed in the spinal cord. So it's expressed in a specific subset of neurons in the spinal cord of mice, and also in horses. We have more beautiful data because you have more reagents and it's easier to work with a mouse. So this is the spinal cord. So these are different populations which are defined by the expression of specific, so PAX7, PAX2. So these are all transcription factors that's important during development of these neurons. And we used an immunohistochemistry, so one antibody with green color for these different guys, and then another color for this DMRT3, and you could see the specific cells, neurons in the spinal cord that during development are expressing this gene. So it's like perfect for being a gene that has a role in controlling locomotion. And not only that, but what we were able to show in this study was that these neurons so they are interneurons, that means that they connect the left and the right side, and they make direct connections with the motor neurons that is, of course, controlling the contraction of muscle during. So what we think is happening is they are controlling limb movement during locomotion in all vertebrates, basically. It's expressed in the spinal cord in other species, including human, and in fish as well. So we think this gene has been recruited to have this specific function. So another example where we start with horse or in a very phenotype in domestic animals and discover a new mechanism which is important in all vertebrates in this case. So the natural gait for humans is in fact a diagonal movement. So you could see that by this. You could see this little girl here. She's walking on. So this is not what. So Uppsala is in fact warmer than Bonn today. It's like 25 degrees in Uppsala today. But in the winter we have it like this. And then she's very careful here to how she moves. And Usain Bolt, of course, will do this diagonal movement. Otherwise, he would never win a gold medal in the Olympics. So this coordination of limbs is super critical for efficient locomotion. So you could, when you leave this today, you could watch yourself how you're swinging your arms coordinated with your legs. So, and if you don't do it, you could let me know, and we could have a sample from you and see if there's anything going on with your DMRT3. So, third and last example, the genetic basis for domestication. And now I will talk about rabbits, in fact. So this is, in fact, a fear. So the thing is with rabbits, is that rabbits, before domestication, they all only occurred on the Iberian Peninsula and in southern France. So all other rabbits that you find in Germany and other countries have been brought here by humans. So they didn't go north because there was too much forest in Europe. But after we cleared forest and made agricultural land, because they like this combination with open land, so to speak. So as I told you, there's been a lot of progress during the last 25 years to find genes and mutations that have altered the phenotype of domestic animals. You have the gatekeeper mutation. This is the rosecomb mutation, which is another mutation we described. And you have graying with age in horses, which is another of the mutations. But we know much less about the genetic changes associated with the initial steps of animal domestication. So this must have involved genetic changes in behavior that allowed animals to survive and reproduce under conditions that would be too stressful for wild animals. Because the big difference between domestic animals and wild is the difference in behavior, how they tolerate humans. They're not afraid of humans. They're often interested of humans. And we use the rabbit, and we think it provides some, and that is because domestication is a relatively recent event. So the wolf was domesticated something like 15,000 years ago, and cattle and so on also like 10,000 years ago. But the rabbit was domesticated about 1400 years ago, so much more recently. And we know where it happened. It happened in southern France. And the thing is that at that time, rabbits only occurred in the Iberian Peninsula and southern France. So they couldn't hybridize with wild populations further north because there were no. And this area is still densely populated with rabbits that we think is very similar in genetic constitution with the ones that were domesticated 1400 years ago. So we could make a direct comparison. So just first to show you this really striking difference between the wild rabbit and the domestic rabbit. So you have the wild rabbit, which has a very strong flight response. And now we could go back to Charles Darwin again. So in the Origin of Species, Charles Darwin writes, "No animal is more difficult to tame than the young of the wild rabbit. Scarcely any animal is tamer than the young of the tame rabbit." So it's very difficult to keep wild rabbits in captivity because they become so stressed, they kill themselves by trying to escape. Whereas the domestic rabbits, you see, you could have it. So this is what we did. So we collected 14 populations of wild rabbits. So these are the sample sizes, sample locations where we have collected wild rabbits. So in fact, there are two subspecies of rabbits, one down here and one up here. And this is the hybrid zone between the subspecies. So we use this also to study rabbit speciation. And then we had 6 breeds of domestic rabbits. And what we're doing here now is that we're comparing base for base domestic rabbits versus wild rabbits. So we wanted to see what is the difference between wild and domestic rabbits. And I don't have time to go into the details because we're running a bit late. But I just summarized the findings. So what we find here is a large number of genes that has contributed to rabbit domestication. So this is what I would say gradual change. It's not, and also this. There was not a single example that the gene has been inactivated. So it's not that you knock out a gene that has to do with behavior and that make them very tame. So it seems that it's a gradual change. Many genes with small effects have contributed to this transition. But what was very striking is that what we primarily saw differences was in genes involved in neuronal and brain development. So we saw very strong signatures that genetic changes around these genes seems to be very critical, which is consistent with strong selection for tameness, as you see here. So what we have done very recently is, in fact, ask the question whether domestication have affected the architecture of the rabbit brain. So the experimental design is that we have 8 wild and 8 domestic female rabbits of the same sex to avoid difference between sexes. They've been kept in captivity under similar conditions. And then we have done high-resolution magnetic resonance image, MRI analysis of whole brain to compare to see if we can see changes in the architecture of the brain. The first finding we have is that the domestic rabbits were on average 4-fold heavier than wild rabbits, but had only marginally larger brains. So these are the blue is the domestic rabbits, these are the wild rabbits. So you see, this is body weight and this is brain volume. And you can see there's in all mammals there's a linear regression between body weight and brain volume. So the larger you are, the larger the brain become. And you would, so what you see is that there's a clear difference between the wild and domestic. So the domestic has a much smaller brain in relation to their body weight than you would expect based on their body weight. And then we went through 54 regions of the brain and compared them, the size, the volume of different regions of the brain. And this was done blind, so the persons that did this classification didn't know if they were looking at a domestic or a wild rabbits. And then we summarized the data, and we were amazed to see that number one, so this is the significance for the difference, but the most striking difference occurs in the amygdala. And then we have also medial frontal cortex, and I will come back to that. So why were we so excited by hearing that the amygdala has shrinked in the domestic rabbit? So this is where you find the amygdala, you find it, so you find it smaller, And the reason is that the amygdala has an important role in memory, decision making, and emotional control, including fear. So it's usually called the fear center. So the fact that we see that it shrinks suggests that it has a role in this shift in behavior. The medial frontal, so it's a specific region in the medial frontal cortex that was also affected, and in fact become enlarged in the domestic rabbit. And this is supposed to facilitate extinction of fear, the process of fear reduction through safety learning, and is generally associated with enhanced emotional control. So the conclusion from this study is that the high-resolution MRI demonstrated the brain architecture is remodeled in the domestic rabbits, showing that genetic changes underlying successful domestication reshape the structural foundation for brain function. And we propose that this reduction in the size of the amygdala and the enlargement of the prefrontal cortex are important reasons why domestic rabbits are less fearful and have an attenuated flight response compared with the wild rabbits. And we think similar changes have happened in other domestic animals, but this has not been studied at this detail at present. But the genetic data and this characterization of the brain goes very much hand in hand, since we see these genetic changes on genes affecting brain and neuronal development. So that is the end of the talk, and I'm happy to answer questions.